today we want to look at hemostasis, and we want to look at blood typing and blood transfusion, and then probably introduce a bit about the heart. Hemostasis. The word tells us what its meaning is. Hemo is for blood, and stasis is for stop. Stopping bleeding, essentially. Hemorrhaging is another word for bleeding. How do we stop hemorrhaging? This would be internal hemorrhaging inside the body, or external hemorrhaging outside the body. What's about our blood volume, approximately? About 5 liters. So you know we can't lose too much before things get serious. So the body has a mechanism to stop the bleeding. And this mechanism is called hemostasis. It involves a 3-step process, but each of these steps is interrelated. It is vasoconstriction, platelet plug formation, and blood clotting. Those 3 are all going to be interrelated. When we look at a typical blood vessel that's bigger than a capillary, not our very smallest blood vessels. If you have a blood vessel, in the lining of the blood vessel, in the wall, we have some smooth muscle. So we have smooth muscle. And we can constrict or dilate the vessel with this smooth muscle. Lining the blood vessels, we have a single layer of endothelium, simple squamous endothelium, which we call endothelium when it lines blood vessels. So we have endo, it's not too good, endothelium, and that lines, puts a lining right on the inside of the blood vessel. Okay? And that's a nice smooth lining. Endothelium. Okay. What happens when we get damage? When there's damage, you get damage in your blood vessel. You expose the blood to tissue underlying beneath the endothelium, which has a lot of collagen. Collagen is the most common protein found in the body. It's everywhere. It's all over the place. It's in our bones, it's in our cartilage, it's in our other connective tissue, and it's in the blood vessels underlying the endothelium. If the blood senses, touches, whatever, collagen, it initiates the reactions of hemostasis and blood clotting. How does this work? Well, let's look at it. When you, excuse me, let me start that again. Inside of our blood, you know that we have the plasma, we have the red blood cells, we have the white blood cells, and we have the platelets. The platelets are just going through the blood vessels, and in their natural state, unstimulated state, they don't stick together and they don't stick to anything else. They're ready to go into action if need be. how do they get called into action? If there is this exposure to collagen, it releases some chemicals which activate the platelets. The platelets release a bunch of chemicals that we'll talk about, and that makes the platelets sticky. They stick to each other, they stick to the lining of the blood vessel where the damage is, and the platelets release chemicals that causes the smooth muscle to undergo vasoconstriction, to contract. What is the advantage of vasoconstriction? Less blood is going to the area of damage. Less blood will be able to leak out. Making the platelet sticky will cause a little platelet plug to be formed, and then other chemicals will start initiating this clotting. So let's talk about these. First of all, here's our vessel damage. That's our blood vessel. That's going to give you an altered endothelial surface. That's that lining of the blood vessel. So your underlying collagen is exposed. That activates the platelets that are just flowing through the blood vessel. It activates them. They're going to discharge a number of chemical mediators. This includes, and we're going to list these for you in just a minute. This includes adenosine diphosphate and serotonin and thromboxane A2. They're going to discharge that. These are then going to cause vasoconstriction, contraction of the vascular smooth muscle. Notice there's some positive feedback here. Blood clotting is one of those things, and hemostasis, where we get some positive feedback. Positive feedback, once you start something, it amplifies it for a while. Oh, we cannot have positive feedback forever. All our blood would clot. We have to have positive feedback only for a time, and then we have to revert to negative feedback. We cannot just continue positive feedback. But if you really want to amp up a response very quickly, you can have positive feedback for a time, and blood clotting fits into that category, where we need to amp it up really quickly. Now, we do normally have two chemicals, prostacyclin and nitric oxide, which keep the platelets away from the endothelial line. But when there is damage, now we see the collagen, the platelets then become activated. The endothelial cells, that's the lining cells, they secrete a chemical called von Willebrand factor. That's a German, so the W is like a V. Von Willebrand factor. That starts the platelets to adhere to the collagen and to each other. The platelets release adenosine diphosphate, serotonin, and thromboxane A2. These cause vasoconstriction, but they also cause the platelets to adhere to each other and to the collagen. Here's vasoconstriction, and here is this stickiness where the platelets adhere to each other and to the collagen. That forms a platelet plug so that blood is not leaking out of the blood vessel as easily. Now eventually that platelet plug gets strengthened into a clot by activating fibrinogen into fibrin, and then we make a clot. That is activated starting here too, but the first two phases, the vasoconstriction and platelet plug, that happened very quickly. It takes clotting just a little longer, but it comes into play as well. This is our blood vessel. We have our endothelial lining. Nitrous oxide, that's what the NO stands for. Here it is down here, nitric oxide. And prostacyclin, which is a type of prostaglandin. They keep the platelets from adhering to the endothelial wall in a normal circumstance. So down here, there's no damage, they keep the platelets from adhering to the wall. But when you have damage, like here, where you have damage to the endothelium and the collagen, which is shown here on the diagram, right there, gets exposed, now you're going to have the platelets releasing these mediators, which causes them to stick together, stick to the collagen, and also causes vasoconstriction. Now, how do we go to the clotting aspect, which is a more longer-term solution, a stronger solution? Well, we have a number of clotting factor proteins. They're really enzymes as well. We call them clotting factors because they help clot. They're proteins and they're enzymes. We have a series, a bunch of these clotting factors, and we can, one by one, take an inactive form of a clotting factor and activate it, which will then act like an enzyme to activate the next in the sequence of clotting factors, which will then activate the next. And finally, we will get the activation of fibrinogen to fibrin, then you form your clot. Thrombin is a clotting factor that directly activates fibrinogen to fibrin. But there's a series of clotting factors that have to come into play to activate thrombin, which will then produce fibrin. Fibrinogen is, you may recall, one of the normal proteins that are in our blood plasma. Fibrinogen is a precursor. To activate it, you have to activate thrombin, you have to activate this clotting mechanism, and then it gets converted to fibrin, and you form the actual clot. All of these clotting factors, fibrinogen, all the other clotting factor proteins, are made in the liver. We're going to be taking inactive clotting factors and we're going to be activating them, eventually forming thrombin, which will convert fibrinogen to fibrin. We're going to see that we have some positive feedback on this step as well. What initiates this? Exposure of the blood to the subendothelial tissue, to collagen. What do we need for these enzymes to become activated? Well, we need calcium to act as a cofactor. We need vitamin K to act as a coenzyme. We need certain other things to help this enzymatic cascade work. So if you're deficient in calcium, if you're deficient in vitamin K, you might have some problems clotting your blood. This is our clotting pathway. When they started naming clotting factors, they gave them sometimes a name, but often just a number, a Roman numeral. Do you know Roman numerals? Is that like, okay, you sort of know what it is? That's getting less common, I think. These are Roman numerals. In other words, this is factor 12. Is that right? The X is a 10, and this is 11, this is 7, this is 9, this is 8, this is 10, this is 5, okay? They're all numbers. Now, it would be so much easier if they numbered these in order of their occurrence when you're clotting. This one does first, and then this one, then this one, then this one. But they named them or numbered them according to when they were discovered, right? Which means they just are random as far as the order.
I've heard that saliva contains clotting factors. Are they these clotting factors?
It can be similar ones to these, and so, yeah, I think you can. But of course, it wouldn't be near notice these clotting factors, they are inactive. If you put a little A behind it, that's the active form. This is the inactive, that's the active, inactive, active, all the way down. When you start with the tissue damage, you start activating it, and eventually you get to thrombin, and then the thrombin down here, if we go back one slide, converts fibrinogen into fibrin. It uses another clotting factor, 13, in that process as well. So this is our sequence, our cascade, if you will. Now, we have two sort of arms of this clotting factor mechanism. We call it the intrinsic factor and the extrinsic pathway. Intrinsic pathway and extrinsic. In the body, they both work together, not just one or the other. They both work together. If you take blood out of the vein, you want to do some study on it, and you put it in a glass tube and it clots, only the intrinsic factor is going, because the extrinsic factor comes from outside of the blood vessel, where the intrinsic comes within the blood, and that's why they get their name. But in the body, both the factors in the blood and the factor right outside of the blood vessel, they work together, and so you activate it. The extrinsic factor, pathway, is faster because it's shorter. But the intrinsic pathway has positive feedback. You see the brown, this is positive feedback. When you make thrombin, you have positive feedback back to these steps. That means it has a more quantitative response, a larger response. But again, in the body, they both work the same. Intrinsic is due to chemicals within the blood. Extrinsic is due to chemicals coming from outside of the blood vessel. Once it gets activated, however, they both work together. We need vitamin K to act as coenzymes in this process. Vitamin K is a fat-soluble vitamin, so we need bile salts to help absorb vitamin K. So it's made in the liver. And then all these clotting factors are made in the liver. So if you have liver problems, you can have some blood clotting problems. That's sort of. Now, once you have the blood clotting, it's not very long before you need to stop it to have negative feedback. How do you stop it so that it doesn't get out of hand? Well, you have tissue factor pathway inhibitor. What is that secreted by? Endothelial cells, those lining cells. This binds to one of our clotting proteins, factor 7, the activated form, and it inhibits it, and it doesn't allow the formation of factor 10. Let's go back. Here's 7A. It doesn't allow the enzymatic formation of 10 because it activates clotting factor 7. Okay? So that stops, slows it down, at least. All right? We have thrombomodulin, which is in the endothelial lining cells, and it can bind to thrombin, which can combine with protein C, and that combination inactivates factor 8 and 5. This is what it looks like. In the endothelial cells, you have thrombomodulin, kind of like a receptor. Thrombin binds to it, then protein C binds to that. That combination activates protein C, which then has a negative feedback on clotting factor 8 and 5. That shuts it down, right? You also have plasminogen being activated to plasmin that breaks down fibrin. One of the reasons we mentioned this plasmin you can give a synthetic form of plasmin or plasminogen when somebody's having a heart attack, so that they don't clot their blood as easily, and so the heart can pump blood easier. Remember, aspirin is also something that helps prevent the platelets from aggregating together, and it's sometimes used if somebody has a heart attack, take an aspirin while you're on your way to the emergency room, whatever, right? To sort of downplay the clotting mechanism. Some people, if they're at high risk for a heart attack or a stroke, take a little dose of aspirin every day, like a baby dose. We call it a baby aspirin. It's usually 8 milligrams. Just a little bit of aspirin every day. And that keeps their platelets from aggregating together. All these clotting factors are proteins, which means their amino acid sequence, as you well know, is coded for by genes in the DNA. If you have a mutation in the DNA, you will make a wrong sequence of your amino acids. The protein's shape, three-dimensional shape, tertiary shape, will be abnormal. The function will be abnormal. Your clotting factor won't work correctly. Two of the most common clotting factor problems, the gene is on the X chromosome. And we know that women have 2 variants of the X and men only have 1. And so men get hemophilia much more commonly than women. It's one of those sex-linked diseases because due to a gene on the X chromosome. We call when we have a mutation so that one of the clotting factors are misshaped. We call it hemophilia. That really means loves blood, right? But anyway, it's a disorder. The most common type of hemophilia is hemophilia A, or classic hemophilia. It's on the X chromosome, and 77% of hemophilia is hemophilia A. Hemophilia B is also on the X chromosome, but it's a loss of factor 9. These account for most of hemophilia. There is a hemophilia C, which is not on the X chromosome, but it's relatively mild because it affects factor 11, and factor 9 can be activated by not only factor 11, but also by factor 7. So there's a couple ways to activate it. So hemophilia C is not as big of a deal. Hemophilia A and B are more of a challenge. Now, if we look back at our here's factor 8. That's the most common. It's misshaped, and so you're not going to get as much clotting. So if somebody's in an accident or they cut themselves or internal bleeding, they may not be able to stop the bleeding, or may have a very difficult time. If you happen to get into the hospital, they can put in artificial clotting factors and stuff, but they might bleed out. Have you heard the story? Probably not. Queen Victoria, way back, she lived, I think she died about 1601 or something. She was a carrier for hemophilia. And back in, all the royal families of Europe intermarried with each other. So she intermarried, her royal family in England intermarried with all the others. And you could go down the lineage and you could find all these males in all these different royal families who were dying from hemophilia. And they traced it back to, they said Queen Victoria was the one that originally had the mutation, and she passed it on. It's thought that the Tsars, which were the Russian royal family. Nicholas and Alexandria had a son, and he had hemophilia, but they didn't know what it was. So he came under the influence of the evil Rasputin monk, who had all these crazy ideas like drowned him partway or something. Anyway, while they were distracted trying to save their son from this crazy disease, the Russian Revolution happened, and there was the end of the Tsar. So, I don't know if you've heard of that, but that's a little linkage with hemophilia back to the royal families of Europe. All right. Any questions or comments or anything you want to chat about on that? Feel okay? Ooh, okay. Let's go to blood typing. You've probably done a little bit of this somewhere along the way. We know that we cannot just donate blood to anybody. Sometimes it's life-saving, but we have to have a match, right? We cannot have a mismatch or we get a transfusion reaction which can potentially kill the recipient. But we have a lot of blood donations, so we know that they have to be of a matching type. So we have lots and lots of red blood cells, 5 million in a drop of blood, lots of red blood cells. If somebody needs a blood transfusion and you're transfusing whole blood with the cells and everything in there, or just packed cells, now you have some potential issues. On your red blood cells, you have some cell markers that we call antigens, okay? These cell markers, or antigens, we have three main types, essentially. Three main types. We have antigens that are type A, type B, and type RH. RH, there are subtypes. D is the most common subtype. Where do we get the name RH from? They originally found this in rhesus monkeys, so that's where they get that from. When we're looking at blood typing, whatever we have genetically, that we have inherited from our parents, whatever blood type we are, we have that antigen on our red blood cells. Our body then recognizes anything that we don't have on our red blood cells as foreign, and we make antibodies, defense proteins, against that. So if they see antigens, they start attacking, if it's a match. So when we're looking at this, if we have a blood type, our antigens would be of a certain type, and our antibodies would be what we don't have for the antigens. So if our blood type is A positive, we have type antigens A, and I'm going to call it D because that's the most common RH. So we'll say D for RH, all right? And the antibodies we have are B. If we have type A negative, we have A, and the antibodies we have are the B and D. If we are B positive, we have B and D, and antibodies are A. If we're B negative, we have B, and then we have A antibodies and D antibodies. And then if we're AB positive, AB positive, we have A and B and D, and we have no antibodies. If we're AB negative, we have A and B antigens. Right? A and B antigens, and we have D antibodies. If we type O positive, we have D antigens, and A and B antibodies. If we have O negative, we have no antigens, and we have A and B and D antibodies. Does that make sense? How that works? We inherit the A and the B and the D from our parents. Okay. There's two genes. We inherit one from father, one from mother. So if we got an A from mom and a B from dad, we could be AB. And then if we didn't have either A or B, we're O. O is a lack of A and B. And then in addition to that, we say, well then, in addition to your A and your B, your A, B, O category, you either have the RH or you don't. About 77%, 70-some percent have the RH or the D, and the rest don't. If you have it, we say you're positive, you're A positive, or you're B positive, or you're O positive. If you don't have it, you're negative, right? Okay, so that's where we get the positives and the negatives. So genetically, we inherit these things from our parents. So who can donate to who? Well, the donor, if you're donating whole blood or packed cells, the donor is donating their red blood cells with their antigens. So you're donating antigens, if you will. The recipient, if they have antibodies that could bind with those antigens, you'll get a transfusion reaction, and it's no good. The only safe reaction is the donor's antigens do not match with the recipient's antibodies, and then it's safe. So, of course, A positive could donate to another A positive because they only have B antibodies that could react. There's nothing to react against, no antigen. A negative could go to A positive. It only has an A antigen. There's nothing for it to react to. O negative is called the universal donor. Why? There's no antigens. So no matter who you give it to, whatever antibodies they have, there's nothing, no antigen for them to bind with and react with. So O negative is the universal donor. If somebody comes in the emergency department and they're passed out and they're bleeding out, you give them some O negative, right? Until you can type across them and make it more specific. So you can easily determine who can donate to who safely if you just remember that the donor blood cells, the type tells you what antigens they have. And the recipient, whatever their blood type is, you look at the antibodies, that's the things that are not the antigens. They're going to be the antibodies that they don't have antibodies. I said that really bad. If you have an A antigen, you have a B antibody. If you have a B antigen, you have an A antibody. Okay, you have the opposite. But that's how we do it. And then you can just simply do that and determine who can donate to safely. Now, the ABO groups, because of the foods that we eat and some of our bacterial flora, we generate antibodies to foreign antigens on our red blood cells before we've ever had a blood transfusion. That means you can't, even the first time, you can't get away with it. But the RH, the D antigen, you can have a transfusion one time with the RH mismatch. Because you don't have a similar antigens in your food, so you don't make any antibodies against it until you get your first blood transfusion. Did that confuse you? Confuse me. it's our first donor, he's never gotten blood. How come it won't work? Because in some of the food he's eaten or his bacterial flora, he's already made antibodies. Because the antigens in the food and the bacterial flora are similar enough to A that he said, that's foreign, I don't recognize it, I'm B positive, I don't. And so as soon as I give him A positive blood, he's gonna react against it. But the positive part, the Rh, the D antigen, is not in food or bacterial flora. He could tolerate that, but he couldn't tolerate the A antigen. He's already got antibodies against it.
Isn't the Rh factor, I remember reading somewhere, like, isn't that the one for people with pregnancies?
Okay, so what's the problem with pregnancies and Rh? That's the problem, all right? Here I say about 85% of Rh, but I said 77, so let's go with 85%. That was a little lower. The Rh antigens are C, D, and E are subtypes, but by far D is the most common. So we often use them interchangeably. What's the problem with a pregnant mother? Well, here's the problem. If the mother is Rh negative, she does not have the D, doesn't have the Rh antigen. She's one of those 15% or whatever. And her husband has the Rh positive, so the baby is Rh positive. Okay? So mother is Rh negative, baby is Rh positive. For the first baby, the first delivery, a little blood generally mixes between the mother's body and the baby's body. Uh-oh. Now the mother has been primed the first time against the Rh factor. Her body says, oh, Rh factor, I'm Rh negative. That's something foreign. Let's build some antibodies. Okay, so she makes a bunch of antibodies. The second and any subsequent pregnancy, if the baby is again Rh positive, those antibodies can cross the placenta and attack the baby's blood and cause a transfusion reaction and could kill the baby. That's the problem. So if you have an Rh negative mother and an Rh positive baby after the first exposure, it causes a transfusion reaction. However, these days, once you know the mother is Rh negative, after every birth, you give a drug called Rhodium, and that prevents the anti-Rh antibodies from forming. And so it's safe if you give Rhodium. And so you can prevent that from happening. So yes, that is a problem. Way back in the day, in the dark ages when I first started teaching, I had 2 different classes with students who were married who lost babies doing this. There was no Rhodium back then. And we would talk about it, and after class they'd come up and I lost the baby this way. I mean, it wasn't that long ago, right, that this was a very serious problem. And if you're in a developing country, they may not have program, or they may not be doing testing, and they may not know the mother is Rh negative or anything, and you could still lose babies this way. So there is a drug that can help prevent it, but there can still be problems. But this was a significant issue back before they developed Rhodia.
Is that how like babies that are like really, really young, like, or like in the womb, how they get like jaundice, or is that like something?
That's usually because their liver is not developing properly. Part of the liver's function, a lot of babies that are preterm, get jaundice, and that's because the liver isn't fully developed, and the liver is supposed to metabolize the bile pigments. And if it's not metabolizing them completely, you get jaundice. This is also why people get jaundice when they have liver cancer or hepatitis or things when they're an adult, because the liver is not functioning correctly. And it's a symptom of that is you're not metabolizing these bile pigments. You have Billy Burton and Billy Rubin, a green and a yellow and you mix it, it's this ugly greenish yellow thing. But what happens if you don't metabolize that, the whites of your eyes get yellow and the skin gets yellowish and it's jaundice, right? And that's usually that's the problem. Now, what you may be referring to is, because hemoglobin can also give color, if you're having a lot of breakdown of red blood cells, that could also probably cause some discoloration. So that may be what you're thinking about. So that could be a potential. But that's very serious, of course, because baby can't deliver oxygen to its cells anymore. And it's a cascade. It starts, all these cells are getting destroyed by these antibodies. And that's the problem. So that's a big challenge, isn't it? Anything else you want to chat about on that? Well, let's move our attention a little bit to the heart. The heart is the next of our three subdivisions of the cardiovascular or the circulatory system. We have the blood, the heart, and the blood vessels. So we're going to do the heart next. Well, when we're talking about the heart, it's a pump. And of course, the human heart is a four-chambered pump. It has two atria and two ventricles. The atria are the little pumps. They just pump right down into the ventricles. The ventricles are the big pumps. The right ventricle pumps to the pulmonary circulatory system, the lungs. It gets the blood reoxygenated, it gets rid of the CO2, and then it comes back to the left atrium and the left side of the heart. The left ventricle pumps to all the rest of the body. It delivers oxygen through our cells throughout the body, and then it returns carbon dioxide back to the right side of the heart and eventually to the lungs. The lung circulation we call the pulmonary circuit, or the pulmonary circulation, and the rest of it we call the systemic circuit. Here's the heart. These are the atria, the right atrium, the right ventricle, the left atrium, the left ventricle. The left ventricle has a thicker wall, a thicker myocardium, cardiac muscle, because it pumps to the high resistant, high pressure systemic system, whereas the right pumps to the low pressure, low systemic, low resistance, pulmonary circulatory system. Over time, we pump equal volumes of blood through the right and left sides, but the left side is the higher pressure, and the right side is the lower pressure. When we look at the valves in the heart, the valves help ensure that the blood is flowing in the proper direction. We have two valves between the atria and the ventricles. These are called, nicely enough, the atrioventricular valves. The one on the right is the tricuspid, the one on the left is the bicuspid or the mitral. The exiting valves from the ventricle are called the semilunar valves. On the right side, it's the pulmonary semilunar valve, and on the left side, it's the aortic semilunar valve. These valves can open and shut, and basically they keep the blood moving in the correct direction so that the blood flow does not go backwards. We have an interventricular septum, which separates the ventricles, because the left side is oxygenated blood and the right side is deoxygenated or less oxygenated blood. This shows you the pathway of blood through the heart. It's important to note that the blood is coming into the right side and the left side at the same time, and it's going out at the same time. But often, just to be descriptive, we describe the blood flow through the right side and the left side, but they're actually going through both sides at the same time. So if we look at this, the blood leaving the left ventricle goes through the pulmonary trunk, the pulmonary arteries, the capillaries of the lungs, the pulmonary veins. You start with deoxygenated blood here, and then after it goes through the capillaries of the lungs and back to the pulmonary veins, it's oxygenated. Then you go to the left atrium, through the left AV valve, through the left ventricle, through the aortic valve, the aorta, all the systemic circuit, back to the right atrium, the right AV valve, the right ventricle, the pulmonary valve, and around like that.
So what you're saying is the most oxygenated blood vessel is vein.
The what?
The most oxygenated blood vessel is vein.
You could say that because the pulmonary veins have, at least as much oxygen as the systemic arteries. We generally say it's the same, but, I see your point. When we look at arteries, a general rule is arteries convey blood away from the heart, and veins carry blood towards the heart. When we're talking about oxygenated versus deoxygenated, it's opposite depending on if you're talking about the pulmonary circuit or the systemic circuit. In the pulmonary circuit, arteries carry deoxygenated blood and veins carry oxygenated. In the systemic, arteries carry oxygenated blood and veins carry deoxygenated. And so there is those differences, yes. One of the main reasons we want to deliver blood throughout the body is we need to deliver oxygen so we can make enough ATP for enough energy. If you're just resting, you're not exercising, your brain is getting, if your cardiac, let me just say, cardiac output is the output of the heart per minute. At rest, a normal cardiac output is about 5 liters per minute. Notice that's the same as our blood volume. We move our entire blood volume through the heart every minute at rest. If we're doing maximal exercising, our cardiac output might be 25 liters per minute. We might move the entire through about 5 times a minute. But at rest, we move our entire blood volume through in about a minute. Where it goes out at rest, about 13% of that goes to our brain, about 4% to our heart, about 20% to our skeletal muscle, and you can see the rest. Kidneys, 20%, because it's filtering the blood. This can change depending on our circumstances. Are we exercising? Have we just eaten? Are we taking a physiology test? So it can change, but this is an estimate. And so we can change that. As we're finishing up here, when the heart is contracting, like the ventricles, we have a name for that, it's called systole. When the heart is relaxing, we call it diastole. This will set us up for tomorrow. So systole, it's contracting, diastole, it's relaxing. By the way, that's where we're going to get systolic and diastolic blood pressure. So I think we covered this. So have a wonderful day.